Good morning again, class. We are now in the continuation of respiration as our topic. Okay. Last meeting, we did ask, or we did have an overview of what a eukaryotic and prokaryotic cell is. Again, when you say eukaryotic, it is a cell with a true nucleus because the nucleus there are membrane bound. And of course, with the other organelles also, they're membrane bound. They are enclosed. While the prokaryotic is a primitive type of cell. Pro means primitive. Okay. Pro means before. Karyon. Karyon means nucleus. The nucleus there are scattered in the cytoplasm. Okay. Instead of having nucleus in the prokaryotic cell, we call it as a what? Nucleolus. Okay. We have nucleolus, which is a nucleus without a membrane. Those are the big difference. Aside from that, prokaryotic cell is usually unicellular. You say unicellular, they can live by their own. Okay, the cell itself is an organism, but a eukaryotic cell is multicellular. Okay, you say multicellular, it cannot live by their own. Okay, so they are actually clustered into group. That's a eukaryotic cell. Okay, making tissues and organs. Please hold on for a while. I'll just fix this one for a while. Let's continue. All right. Those are their similarities and those are their differences. Right. The primary function of the mitochondrion, again, mitochondrion is singular, mitochondria is plural, is to produce large amount of energy in the form of adenosine triphosphate. That is the energy currency of the cell. Okay. For various cellular functions. Remember that without ATP, we cannot perform metabolic processes because it is directly involved in the metabolic processes. When you say metabolic processes, these are the processes of our body. Okay, metabolism. Mitochondria are generally round shape, ranges from 0.5 to 10 microns. Mitochondria are present in eukaryotic cell. However, they are found in high concentration in animal muscle cells because our muscle cells needs a lot of energy in order to perform its function, which require more energy. Mitochondria make as much as 60% of the volume of muscle cells and 40% the volume of heart cells. Other than generating energy for the cells, mitochondria also involved in other functions such as signaling cellular differentiation. It is actually involved in cell differentiation. When you say cell differentiation, like for example, have you observed a zygote? Have you seen a zygote? Okay, even in the video, you didn't actually observe zygote,
I have,
the
sir, but I
developing
don't
baby.
remember.
Okay, the developing baby is just a single cell. Question, how can we actually get the different parts of the body out of a single cell? Remember that it is a product of the union between egg and sperm. If it is a product between a union of egg and sperm, therefore it will develop a single cell because it is a union. They united. Okay. So if it is a union, we actually created a single cell. Question, how can you get in different parts of your body? How did you get the different parts of your body out of a single cell? Anybody? Robbie, EM, Jillian? Janine? Vincent, Kyle, Hillary? How? How can you get all of those parts? This is because of, are you still with me?
Yes.
Yes.
Okay. This is because of cellular differentiation. This is not the process of cellular differentiation. And the one who gives signal for cellular differentiation is your mitochondria. Okay? This is cellular differentiation. It can create or it can actually generate different cells out of a single cell. Okay? Right. Cell death as well as control of the cell cycle and cell growth. Okay? Like chloroplasts, mitochondria also possess DNA or the deoxyribonucleic acids and ribosomes from different DNAs found in the nucleus of the cell. Right? Mitochondria are organelles with a double membrane structure. You know already why it is double structure. Okay? Which made up of phospholipids and proteins. Okay, the outer membrane defines the shape of the mitochondrion. It is completely permeable for nutrients, ions rather, and ATP and ADP molecules. ATP again is adenosine triphosphate, and ADP is adenosine diphosphate. This is the used up ATP. The inner membrane is more complex structure. Okay, it is made up of large number of proteins and play an important role in producing ATP, and it also contains complexes of electron transport chain, the ATZ. Later on, we'll be discussing about ATZ. We'll be discussing about glycolysis. We'll be discussing about Krebs cycle and fermentation. It is permeable only to oxygen, carbon dioxide, and water because, again, these are the raw materials for respiration. Okay. The inner membrane has a folding called cristae, okay, that increase its surface area. Okay? Many of the chemical reactions occur in the inner membrane. The space between the outer membrane and the inner membrane is called intermembrane space. It should have a space in order for it to what? To move. Okay. There are a lot of chemical reactions happening in the mitochondria. Okay, we're still in the mitochondria. Due to the presence of electron transport system, or ATZ, in the inner mitochondrial membrane, the intermembrane space contains high proton concentration. Okay, the matrix is a complex mixture of enzymes that are important for the synthesis of ATP because ATP synthesis happens actually in this area, in the matrix. ATP molecules, special mitochondrial ribosomes, and mitochondrial DNA. This is the reason also why the tail of the sperm, once penetrated in the egg, will just separate by itself because it contains DNA. Again, mitochondria contains DNA. That is the mitochondrial DNA. So it needs to be separated. The tail of the sperm should be separated before penetrating or after penetrating rather in the egg in order to contain what? The genetic characteristic of the sperm. Okay, if you're going to look at the structure of mitochondrion, you can see that there are some interspace there Okay, the interspace plays a vital role in synthesizing ATP. ATP synthesis happens here. We have the cristae. Okay, we have also ATP synthase. Okay, the DNA, because mitochondria has its own DNA, mitochondrial DNA, and of course the two membranes that actually covers the entire mitochondria. Enzymes. Enzymes are proteins that accelerate chemical reaction. Okay, it catalyzes these reactions. Specific enzymes accelerate each reaction. In cellular respiration, okay, the role of enzyme is to carry electrons from one molecule to another. Okay, again, the role of enzyme is to carry electrons from one molecule to another. That is in the cellular respiration. Okay, the movement of electrons between molecules represent an oxidation reduction reaction. Okay, oxidation reduction. Okay, where the loss of electrons from one molecule, that's the oxidation, we lost an electrons, corresponds to the gain of electrons in another substance, that's the reduction. Okay. Coenzymes are non-protein molecules that work with the enzyme in various chemical reactions inside the cell. Later on, we'll be discussing about this also. But in the cycle, when I created the illustration of the cycle, I didn't actually include all the enzymes simply because they're too broad. At the same time, you cannot actually familiarize them, and it make your idea more vague. So it's difficult if you're gonna add up more. I will just put it I will just put a complete set of those in your higher years. Okay, I can. But as of the moment, this is just an introductory lesson. It is an introduction of the lesson. Okay. These are the coenzyme. You're going to look at the coenzymes. We have the NAD, that's nicotinamide adenine dinucleotide. Okay, this is involved in the redox reaction. Okay, carrying electrons from one reaction to another. On the cell of two forms, you have NAD plus, which is an oxidizing agent, accepts electrons. If it accepts electrons, okay, it will become NAD PH. Okay, NAD H, NADH, which is a reducing agent, donates an electrons. It actually donates electrons. Okay. The next one is flavin adenine dinucleotide. Okay. A redox coenzyme. Okay. Organic non-protein molecule that helps enzymes catalyze a reaction. It exists in three forms. Okay. The FAD, FADH. Okay. Nicotinamide adenine dinucleotide. Then FADH, FADH2. So these are their forms. Okay. Acetyl coenzyme A or acetyl CoA. Okay. A carbon atoms. Okay. It carries a carbon atom within the acetyl group to citric acid cycle or Krebs cycle to be oxidized, okay, for energy production. Later on also, you will understand the functions of these coenzymes because it is included in the illustration. Stages of cellular respiration. Okay. Respiration or cellular respiration, this is the way how we actually create energy. At the same time, this is how we synthesize ATP. Okay, carbohydrates, fats, and proteins can all be used as fuels for cellular respiration, of course, because these are stored energies. But glucose is most commonly used as an example to examine the reaction and pathways involved. We need to isolate. That is why we use glucose in the process, because these carbohydrates, fats, proteins will be what? This will be broken down into smaller pieces in the simplest form, which is the glucose, the C6H12O6. Okay. ATP production in cells can take place either through the aerobic or anaerobic phase or processes. You say aerobic, it involves oxygen in the process. Like in our case, okay, we are aerobic organisms because we are actually using oxygen in the process. You're actually using oxygen to do our metabolic processes. Okay? Anaerobic process or anaerobic processes means that the organism or the process itself didn't actually maximize oxygen. Okay? It doesn't need oxygen in order to perform its metabolic processes. Like for example, anaerobic bacteria found in your colon. They didn't actually use oxygen. Okay? But they are actually performing their metabolic processes. Okay, what else? Bacteria found in fermented fermented juices. Okay, this is how we create also your suka, your vinegar. Okay, those are anaerobic processes. Okay, let's take a look at aerobic respiration. Okay, aerobic respiration requires oxygen in order to generate ATP, while again on the other hand, anaerobic respiration does not require the presence of oxygen in the process. Okay, the process can be illustrated as, okay, C6H12O6, this is your sugar. Remember, take note, this is your glucose. This is the chemical formula for glucose, the simplest form of sugar, plus 6 oxygen, okay, yields to 6 CO2, 6 carbon dioxide, and 6 H2O. So the product of respiration is, again, 6 CO2 and H2O or water. Okay. Cellular respiration can be divided into 3 metabolic stages. Okay, you have glycolysis, citric acid cycle or citric acid cycle, and electron transport chain. Okay, again, glycolysis, citric acid cycle or Krebs cycle, and electron transport chain. Okay, both created, or they actually created, sets of ATPs. Later on, we will also we'll be also tracing the pathway of these ATPs. Glycolysis. Glycolysis occurs in the cytosol. Okay, the cytosol is the actual place for glycolysis to happen. In glycolysis, the 6 carbon sugar, glucose, okay, the C6H12O6, is broken down into 2 molecules having 3 carbon atoms. Okay, each resulting molecule is called pyruvate or pyruvic acid. Okay, this transformation is accompanied by the net gain of 2 ATPs or 2 ATP molecules and 2 NADH molecules, so NADH molecules. So if you're going to ask the process, this is the entire process of glycolysis. Okay, the first phase will be the investment phase because we're gonna use ATP in the process, and the second phase is the profit phase because we actually gain ATPs in the process. Okay, so let's move. Okay, the first phase of glycolysis is the investment phase. It started with the glucose, correct? The C6H12O6. Okay. Now we use 1 ATP in the process to create glucose 6-phosphate. We added phosphate in the group. Okay, glucose 6-phosphate, meaning to say that the phosphate is added in the 6th, ika-unum, 6th carbon atom. Okay. And in the process, it will actually convert it into fructose 6-phosphate. We also use ATP in order to convert it into fructose 1,6-bisphosphate or biphosphate, bisphosphate. Okay, again, we need 1 ATP in order to convert it into fructose 1,6-bisphosphate, meaning to say that in this 6-carbon molecule, okay, phosphate is added or phosphate is being attached to fructose 1, meaning first atom, first carbon in this chain, first carbon, and sixth carbon. Okay, we added actually a phosphate in the first and the last. Because we added in the first and the last, we can now actually generate glyceraldehyde 3-phosphate. Okay, so we actually created 2 G3P in the process. Okay, we created 2 G3P in the process. That is why in the second phase, it will happen twice. Okay, the second phase will happen twice. Sure, why the second phase will happen twice? Simply because we created 2 G3Ps or glyceraldehyde 3-phosphate. Okay, 1 and 2. We divided actually the C6H12 into 2 G3Ps or glyceraldehyde 3-phosphate. Okay, let's move now to second phase. Okay, the requirement or the raw materials for the second phase is your G3P. Okay, let's isolate one. Okay, let's isolate one G3P, this one, and let's move to the profit phase. Okay, in the profit phase, we have glyceraldehyde 3-phosphate, meaning to say that the phosphate group is added in the third carbon molecule. Okay, we converted, okay, NAD into NADH in the process to create, okay, to create 1,3-bisphosphate glycerate. So we use actually the phosphate group in the other end. Okay, it is connected in the phosphate group is connected in the first carbon molecule and the last carbon molecule or third carbon molecule. Okay. In the process also, we created, because this is the profit phase, we created ATP. We converted ADP into ATP. So we have 1, okay, 1 NADH and 1 ATP, okay, to form 3 phosphoglycerate. Okay, this is a 3-carbon molecule with attached, okay, phosphate group in the third carbon molecule. Okay, then the 3 phosphoglycerate will be converted into 2 phosphoglycerate. When you say 2 phosphoglycerate, the phosphate group is attached to the second carbon molecule. And there you go. It will be converted into